Okay, for the second part of the first chapter, we're going to go over broadly what ecology is, give a couple examples, and talk about scale and the interaction of the abiotic environment. So to start that off, we'll go with a few definitions. Ecology referring to the scientific endeavor, not environmental activism. And then environmental science incorporates concepts from the natural sciences and social sciences and focuses on how people affect the environment and how we address environmental problems. And obviously, you are in an environmental science program, and I think we strive to fulfill this definition. Huxley was created to really address this problem, and over our history, we've bifurcated to environmental science and environmental studies, but we hope to still fill that general definition of environmental science through a mixture of coursework that includes environmental studies courses or environmental science courses that do incorporate what we call like the human dimensions, but realizing that our environment is heavily influenced by people. And actually, in fisheries management, it's interesting. A lot of my friends acknowledge that fisheries management isn't about managing fish; it's about managing people, right? Like the fish will do their thing just fine. It's the human influence, either through, as we'll learn about in the next chapter, habitat destruction or other impacts that impact fish more so than managing the fish, like telling the fish what to do. It's telling the people what to do. So similarly, that's the idea of environmental science compared to like pure ecology. In our view of ecology has changed. Early views are there's a balance of nature, which natural systems are stable and tend to return to that balance after disturbance. And each species plays a distinct role. I think the balance of nature has really shifted really more to from a sustainability view to a resilience view. So natural systems have multiple stable states and they can bounce between different stable states. And so as ecologists, we now recognize that natural systems don't necessarily return to their original state of disturbance, and seemingly random perturbations can play an important role. George Sugihara has done really good work on that around fisheries and the introduction of chaos in those in fisheries modeling, meaning using East Coast cod as an example. Cod were greatly overfished and depressed, and they haven't immediately bounced back because then something else can be at play, meaning in that case maybe lobsters are taking over and the system is shifted from a cod-dominated system to dominated by something else. And so current evidence suggests that different communities can form in the same area under similar environmental conditions. Again, this is hinting at this idea of resilience in alternative stable states. One example I give of that are coral reefs. And so you can imagine a beautiful, pristine coral reef, bright, shiny, happy corals, lots of colorful fish. But if enough of the keystone species are removed, combined with the addition of nutrients, that coral reef will quickly become covered by seaweed and no longer be a coral reef, now just be in an algal dominated system. Two totally different states, and they're relatively stable in their own right. So yeah, the world is now more complex than we thought, but we still need to fundamentally understand that the world's interconnected, and the sort of like test tube beaker scale experiments are really important when they test a specific question formulated and hypothesized based on complex interactions we see in the natural world, not vice versa. And that's, I think, the really cool thing about the frog example is it kind of walks through that observation in nature, replication using natural gradations, and then moving on to the mechanisms that are really better done in the lab, white blood cell counts. So, a number of definitions. You should be familiar with all of them and be able to define them per the textbook. We're going to walk through them all, so I'm not going to recite them here. Let's see, let's move this. So, ecologists must think of appropriate scales. And this is kind of going back to a bit on the frog example, perhaps not, but we can think of small scale interactions, what's happening with mycorrhizal fungi, and how it relates to plant distributions in a meadow. And then we can also look at how does atmospheric pollutants or deposition impact the color of snow fields, which then impacts the albedo and the rate at which they melt. Temporal scales, similarly, there's short scale, growing season, daily, weekly, but then long species change over geological time is probably a bit longer than we would typically do. But the temporal scale piece, I think, is really important, particularly as it relates to something called shifting baselines, which is Daniel Pauly's work. I think it's a 1996 paper out of Tree. Really changed the way that people think about ecology and systems, which is to say, in his shifting baseline story, when you first are exposed to an ecosystem, you set that as your baseline. So the first time you walk through Seaholm Hill, you think this is what Seaholm Hill is supposed to look like. And if you come back 20 years later, you're going to say, oh, it's changed. So from your baseline, it's changed, but that's not you're unable to really comprehend or take in what did Seaholm Hill look like 200 years ago, because you weren't there, so you don't really have that baseline. And so the arguments that every generation that comes through and studies an area, they first come to that area, that becomes their baseline. They see degradation over time. The next generation comes in to that now degraded ecosystem, sets that as their baseline. It degrades over time. The next generation comes in, sets that as their baseline. They see degradation. But each individual generation sees a gradual change, but they're unable to see that change over decades or hundreds of years. And because of that, we've accepted a decline in our ecosystems that if we had the full view of, we would not. So again, it's called shifting baselines. We'll get into it later. But it's really, to me, like one of the fundamental things that's changed my career. So looking at things over long time scales, and most of my work's kind of been at historic ecology, not necessarily geological time. Most of my work is in the Holocene, going back a couple thousand years, but not on the order of tens of thousands. And so looking at interactions across many levels of organization, and we can either focus on the individual, population, community, or ecosystem scale. And what that's really saying is, I think this is the next slide. Yeah, the next slide illustrates that, which is we can study an individual fish. We can study how fish interact. And then here is a picture with, depending on who you believe, many different fish or many fishes. Per Ted Peach, who's written multiple books, is a well-known ichthyologist from University of Washington. His standard is, here are some fish. Here are some fishes. Multiple individuals of the same species is still called fish. Multiple species together, you can use the word fishes. And technically fish is also still correct. And so that's the guideline I go by. So here I see a fish, here I see two fish, here I see many fishes, because there's at least two different species there. Okay, so individual, multiple members of the same species, multiple species, including invertebrates and the other and their habitat, coral reefs. Then we can zoom that out to overall island ecosystems or coastal ecosystems, and then move that to globally. So to be an ecological study, you can look at an individual, but you also have to have that question grounded in a broader context. Meaning, similarly to the frog white blood cell count study, we can alter an organism to see what happens, but that question is driven out of a larger observation based in the natural world. And to help define those questions, we can use a number of terms. A population being a group of individuals of a single species that live in a particular area and interact with one another. Meaning, if I say a population of grizzly bears, I generally don't mean every grizzly bear in the world. I generally would define that to a discrete area. Central coast, coastal grizzly bear populations. So now we're talking about grizzly bear populations in the central coast, so the center part of British Columbia on the coast. And then the community is an association of populations of different species in the same area. So we can talk about the Central Coast riparian community. So we've got an area, the Central Coast of British Columbia, and a subset of that is the riparian area, and then the species around that. And ecological studies frequently include both biotic and abiotic, meaning biology, living things, and abiotic things. Rocks, minerals, water, temperature, rain, stuff like that. An ecosystem is a community of organisms plus their physical environment, so including the environment. And landscape is an area of substantial differences, typically with multiple ecosystems. And so we could talk about landscape scale ecology and maybe looking at the Western Cascades, something like that, which would include a variety of different ecosystems: riverine, estuarine, plain, mountain, alpine, things like that. And all the world's ecosystem comprised the biosphere of all living organisms. Very few studies are biosphere scale. There's like the Costanza paper that puts an economic evaluation on the Earth. The Halpern paper, which looks at human impact across the globe and argues that every aspect of the globe has been impacted by humans, either directly or indirectly, i.e. via pollution and atmospheric deposition. But those are like kind of large, like meta-analysis modeling type studies. And we can talk about evolution, which is the driver of a lot of the differences we're seeing. So it's a change in a genetic characteristic of a population over time. It's a descent with modification. Organisms gradually accumulate differences from their ancestors. Meaning there has to be... Sorry, my kids are running around. I'm not sure what's going on. That there has to be a inheritable modification. We should all be familiar with, generally evolution at this point in time. So as adaptation, a characteristic that improves survival or reproduction, there has to be something that gives you an advantage. And then natural selection, meaning the individuals with that advantage tend to have more offspring, and that adaptation is heritable. You can pass it off to your offspring. This is kind of a funny example. This is looking at bacteria and an antibiotic. And so here you've got a suite of bacteria This sieve is to represent the antibiotics. And so we've got this suite of bacteria. I would say 10% are the blue ones. I'm colorblind. I think they're blue. They could be purple. Who knows? And 90% are these kind of reddish ones. They all go through the sieve, and they come out where a third of them are the blue ones and 2/3 are the red ones. They reproduce in about that same order. Go through the sieve again. Now we've got 2, 4, 6, 8, and out of that, 2 are red. So now 3/4 are blue and 1/4 are red. So in this case, the blue ones represent antibiotic resistant bacteria. So a few of the non-antibiotic resistant bacteria make it through just due to random chance. The bacteria is not 100% effective, etc. But the blue ones are totally resistant, so you can keep hitting it with the antibiotic and they're totally happy. Generally, what that does is it shifts our microbial communities to being antibiotic resistant. These superbugs, things like that you get when you go to the hospital, is kind of the foundation of that idea. And we've got, generally, ecosystems are structured with producers and consumers. Producers or autotrophs use energy from an external source, typically the sun. Of course, there's a number of other examples, including chemosynthetic pathways, methanthropes, bacteria or archaea that live in the deep sea that feed off hydrothermal vents, off the hydrogen sulfide coming out of it, or the methane coming out of the deep sea. But generally, most of our primary production comes from the sun. And we can talk about net primary productivity, which is the energy captured by producers minus the amount lost as heat in cellular respiration, which means that a plant fixes some carbon, but it needs some amount of that energy it fixes, the sugars it fixes, to run its daily business. And so the amount left over after the plant pays its bills is net primary productivity. And the interesting thing about that is frequently we talk about blue carbon and like kelp forests reducing ocean acidification, which is true during the day when they're taking in carbon dioxide and fixing it. But then at night, they're still respiring, right? They still respire and release carbon dioxide. So actually at night, it can turn it the other way. And then consumers are heterotrophs. They eat things that are that have either made their own energy or gotten energy from somewhere else. So they get their energy by consuming other organisms or their remains. And all energy captured by producers is eventually lost from the ecosystem as metabolic heat. So all of the energy fixed is ultimately lost through consumption and then being burned, released as metabolic heat. So energy that moves through the environment goes in one direction. Typically, energy is fixed from the sun in a plant. It's consumed through the animal. It uses some of that to provide energy, which ultimately generates heat. The rest is passed out the back end. There may be some energy left there, but it's consumed by bacteria, etc. But primarily what comes out the other side are nutrients, and those nutrients can be continuously recycled. So the nitrogen, phosphorus, etc. are typically able to be recycled. And that's called the nutrient cycle. And the nutrient cycle does have some leaks, but it's typically more of a recycle system where when we think about energy, it's a one-way flow. So it's a pretty big difference between the two. Here's an example of that, of incoming solar radiation, producers, and the energy flow going downward of critters that live on the plants, birds eating them, etc. And then the energy flow sort of ending, where the nutrient cycle is a cycle where here are grasshoppers consuming the grass, defecating, that's feeding some other grass, and it's kind of going around and around. So it's the big difference there is a cycle versus flow. So energy flow, nutrient cycling. Those two words should give you an indication of what's happening. And there are many different ways of approaching ecological questions. There's people who do really pure observational studies in the field. You could think of the person wandering around in the woods, listening to birds, and that makes sense for that sort of question, right? You can't take a bunch of birds, bring them into the lab, and ask them, "Hey, where do you like to live?" Like, you have to go out there to see where they're at. Then there's controlled experiments in the laboratory, which could be based on observations from the field. Ideally, they are. And then there's field experiments where we're manipulating the field. Exclusion experiments, things like that are pretty common. And then we can take existing data and create quantitative modeling. And so either from our experimental data or from existing data, and build models to make predictions about how we think the environment works or how things will change. This diagram shows it quite nicely, where you've got beaker scale experiments, and you've got mesocosm scale, which in this case are a bunch of cattle tanks. So you can have a bunch of different water samples here and add different amounts of, say, nitrogen, and see what happens to the phytoplankton growth. Or you can do it here in a larger scale, or in the example of the, it's not the Great Lakes. There's a lake center in Canada. It's where the Schindler paper was done. Something Lakes experimental area, where there's a bunch of lakes where they can do large scale experiments. And the Schindler study, which we'll talk about through the quarter, was one where they roped off half of a lake, and they added nitrogen to both sides, and one side phosphorus, and the side with phosphorus had a massive amount of phytoplankton growth. So it's a way of trying to get at what's the limiting factor on an ecosystem scale, and we can do that in the beaker scale, and we can do that in the bucket scale. But it's really easy for people who are opposing that data from impacting regulation to say, well, that's cute, but it doesn't impact the overall ecosystem. Like, you can do something in a bucket, but it doesn't mean it would impact the overall lake. And so these large scale experiments are very difficult, very rare, but also very valuable if done correctly, because they provide profound data that's hard to argue with in a regulatory management scheme. So sometimes experiments are difficult or impossible to perform, long scale, long time. And so we often use, like this global warming example, a mixture of observational, small scale, and quantitative models. So in the kind of global warming, climate change umbrella, you can think about ocean acidification studies. And there's ocean acidification centers that have a variety of tanks of different sizes where they can crank up the CO2, change the acidity, and see what happens to things like oyster larvae. We don't do that in Puget Sound. We don't just go through a whole bunch of carbon dioxide in Puget Sound and see what happens. And so we have to try to replicate those conditions in a controlled system, and based on what happens, then use those data to model what our future is going to look like. We're just not able to do manipulations at the scale required to give us a full glimpse of the future. We kind of have to do it in various, actually a lot of them are like those 120 quart coolers, but in various kind of small bucket scale stuff. And climate change is a directional change, kind of have a little issue with directional change, but it's change in climate because it can increase variability as well. That occurs over 30 years or longer, and climate impacts all of the things we see. The next chapter is devoted more to that and the kind of abiotic impacts, as well as some climate variability that's really super interesting as well. And so in experimental design, we'll talk a lot about replication, and we talk a lot about random. So replication and random are two things we're always striving for. If we just have one control and one experimental, it's hard to say, are those results by accident, or is there something actually there? So we try to replicate, like a good rule of thumb is like at least 10 replicates. Depends on the study and the scale. And that's where when we're looking at things like if we want to manipulate a large area, over hundreds of square meters, doing that 10 times for your control and 10 times for your treatment becomes difficult. So sometimes we'll sacrifice replication for scale, or we can keep our studies much smaller and have a lot more replication. And we've tried to keep things as random as possible. So you can imagine like the lawn in between Huxley and Communications and Academic West. I think that big kind of grassy area where the stairs to nowhere are. Let's say we want to do a study looking at the impact of fertilizer on dandelion growth. So we say, well, we're going to take the left half of the field and put fertilizer on it, and the right half and not. Well, there could be differences between the left and the right side of the field, or the east and west. Is probably a better way of thinking about it. And so we're not accounting for that. And so really the best way to do that is to break it up in little patches. And the next example will show that. And then have a bunch of different patches, assign them numbers 1 through 20, and randomly pull out of a hat. Number 1, you can be fertilized. Number 2, you're not. Number 3, you're not. Number 4, you are. And randomly assign treatment or control to that distribution of patches. And we try and do all this so we can use statistical methods to say whether or not our results are significant. And so the number one thing you see, or the number one thing like faculty will work with students on is they'll say, oh, it's significantly more. It's like, well, how? Like, how do you know that? You can't just say the word significant has significance. The word significant needs to be backed up with a statistic that conveys that information, which is why replicates are really important, because the more replicates you have, hopefully the more powerful statistics you can use, and the smaller your variance gets. And so you can measure 2 controls and 2 treatments and have a widely different value for each of them. But if they have huge error bars, then they're going to overlap, and you can't say there's anything significant about it. But if you do enough to where you can reduce those error bars, then you can start to have a significant result. So here's an example of that kind of field analogy I was talking about between Huxley and Academic West, etc., where they're interested in a bunch of different plots and then applying nutrients to these different plots. And so they have 5 by 5 meter plots, and they have a whole bunch of them laid out in a grid. So they laid them out in a grid, and they mowed the lawn, and they said some of you will get treatments and some of you won't. They randomly assigned treatments and non-treatments in equal numbers, and then went to see what happened. And so here's the investigator, and then this is on the right sprayed with insecticide, and on the left it's a control plot. So here they sprayed insecticide, so that assumption is with the lack of herbivory, the lack of insects chewing on the plants, they grow bigger. When you have insects chewing on plants, they don't grow as big. And to do that, they took this big, huge field and sprayed different areas. And it's kind of cool. You can see it from here, where there's like some that are quite a lot larger than others, like kind of right away. And then here with the author for scale, you're like, okay, that's more plants. If you kill all the bugs, plants grow bigger. And so ideally, we're going through this iterative process and putting our faith in the scientific method, where we start off with observations and then ask questions. We try to be familiar with ecosystems and have some level of natural history, where we understand how organisms at least interact on what I'll refer to as first principles. And so first principles are things like water is wet. clams eat phytoplankton, like first principles, like just the basics of like if you had to sketch the ecosystem out on a whiteboard, like what would you include? Um, and and then survey the literature to use previous knowledge to develop hypotheses, evaluate those by experimentation or observational studies, and then look at your results and either modify your hypotheses, pose new ones, or draw conclusions, and then put that out there for it to be further refined. And so the idea is one of my advisors would talk about, putting a brick in the wall of science, that we're hoping to produce a piece of knowledge that goes in the brick of wall of science. And perhaps some discovery will come that will crash down that entire wall, and then people will start putting bricks into that new wall. But that's ultimately our goal, is to provide a piece of information to better understand the world as ecologist. So we can return to this example of the frog, which is really pretty interesting because it does bring in a whole bunch, both just like movement of an observation by kids of like frogs with multiple legs, like pretty freaky. And then, these ideas, maybe it's the cyst, and being able to have some results there, and taking it further to looking at these compound impacts of insecticide or pesticides, rather. Sorry, pesticides is a broader term. So these pesticide impacts that are amplifying the parasitic worm impact on this misdevelopment of the frogs. And so it's really pretty interesting, where there's like a lot of different pieces going on that took a lot of work to suss out. It wasn't just like a slam dunk right off the bat, like, oh, I can see how all these 10 things are related and like clearly answer it in a one-off. No, it's very complicated. And the first piece of information led us to the second, which led us to the next, which led us to the next. And so that's what we mean by these iterative processes, that science by nature is iterative. As you embark on a new study, likely you fail spectacularly the first time, and that's part of it. That is part of the iteration of doing a thing and then it failing. And hopefully you learn something from that, both about your methods, and you can take that and design a better experiment. So this chapter kind of lays out the foundation of kind of where we're going in ecology, introducing some terms and concepts, many of which I hope are refreshers. And then from here on out, we're kind of taking it 2 chapters a week and working through the book. So this is, again, just a pretty broad overview, so we're all speaking the same language and have a refreshed understanding of what ecology means and how it functions and how it's done kind of on a day-to-day basis. With that, thank you.